So good morning. First of all, I would like to thank the audience for attending this webinar. Today we have the webinar of the Metabolism and Nutrition group that is coordinated by Josep Villena. Thank you, Josep, for organizing this webinar. And please, if you want, introduce the Glòria to the audience. Thank you.
Hi, thank you. Mar, as Mar said, I'm Josep Villena. I'm a researcher here at the Vall Research Institute here in Barcelona. I just want to say that that's my first time in one of these webinars of the Metabolism and Nutrition group of the SEBBM. And before introducing our speaker, I just wanted to thank the former coordinator, Glòria Garrabou, for her task at front of this group. So today we have as a speaker Glòria Garrabou. She's a researcher at the IDIBAPS, the Institut d'Investigacions Biomèdiques Agustí i Sunyer here at Barcelona, which is located at the hospital clinic. Okay. And particularly, she's a researcher of the muscle research and mitochondrial function group, and also a member of the FIBRoblasts. She's a biologist, and well, she's been working for a long time on neuromuscular and neurological diseases, and using particularly cell models that model this kind of diseases. And I believe that she's been doing it from a point of view of mostly of bioenergetics and metabolism. So that's the reason she's here today with us. So the title of her talk is Omic and Functional Validation of Bioenergetic and Metabolic Reprogramming in Fibroblast Models of Neurological and Neuromuscular Diseases. So Glòria, that's all yours. So thank you for being here with us.
Thank you so much, Pep, Dr. Villena, for the introduction, and good morning, everyone. First of all, I would like to thank the organization, not only restricted to Pep, Dr. Villena, that for sure is the amphitryon of this talk, but also to, as he said, to Glòria Garrabou, that was the previous organizer, because both introduced me or invited me to participate in these seminars. It's my pleasure being here, and I would also like to thank all the technical stuff that was done by both Mars, Maria del Mar and Mar, without whom help I wouldn't be here connected. In this talk, I would like to focus on cell models, as Pep said, on neurological neuromuscular diseases. We will explain how we validate these models of disease, and also in the meanwhile, while we perform the validation, how we depict molecular key players that may be used as biomarkers or even as therapeutic targets, and for sure to know the etiology of these disorders. And I will try to be concise and not extend on the contents of this talk in order to save time for questions and discussion. So let's start with the speech. I will try to share the screen. Hopefully you are seeing my screen. Is it working?
Yep.
Yes.
Okay. So let's start, or let's introduce the diseases we would talk today that are neurological and neuromuscular disorders. They differ in the the affected target tissue, as well as in the clinical manifestation of both disorders. These are signs and symptoms that experience the patients, but they also share some similarities. For instance, the huge sociosanitary burden associated to both diseases, as well as the huge amount of patients affected by these disorders, at least in developed countries, because they are both diseases associated to aging, and this is why they are more prevalent in first or developed countries. They also are highly discapacitating diseases. In the case of neurological disorders, they are usually associated to the impairment of high cognitive functions, but also to movement disorders. In the case of neuromuscular disorders, they are mostly related or restricted to locomotion impairment. Both can be classified depending on their etiology, on which clinical signs and symptoms are characteristics, as well as which part of our body is affected by the disease. In the case of neurological disorders, the central or the peripheral tissue, and in the case of neuromuscular diseases, either the nervous system, in this case the motor neuron, the muscle tissue that is innervated by this motor neuron, and also the motor junction between both entities. So any of these elements can be affected in the case of neuromuscular disorders. Neurological diseases are more prevalent than neuromuscular disorders that are usually rare diseases, but they affect a lot of million people worldwide. We will focus today in the study of Parkinson's disease and inclusion body myositis because they are quite prevalent diseases in their respective classifications. In the case of Parkinson's disease, is the second most prevalent neurodegenerative disease worldwide, and inclusion body myositis, abbreviated as IBM, is the most prevalent myositis in patients over 50 years. So they affect a lot of people. And we also focused in these diseases because they are highly related to metabolism. That is the focus of this seminar. So we will later talk about this relationship. The typical pathognomonic features of Parkinson's disease at clinical level is the presence of mobility and movement disorders like rigidity, tremor, bradykinesia, akinesia, or postural disturbance. These were the main symptoms. But in the late years, we have discovered that this disease is also associated to the presence of constipation, sleep disturbances, depression, hypotension, or sweating, among others, that usually appear before the pathognomonic signs of the disease. So they can be used to diagnose the disease in the prodromal stage where the movement disorder is not still present. At molecular level, this pathology is due to the death or dysfunction of the dopaminergic neurons in the substantia nigra pars compacta of the central nervous system. And these neurons are responsible for movement, production or induction through the release of dopamine. And when these neurons do not work, we experience Parkinson's disease. In the case of inclusion body myositis, this disease is restricted to muscles. Typically, the quadriceps and the finger flexors are the first muscles that are affected by this disease. And this disease is characteristic of affecting distal and proximal muscles, and usually manifested by the drop of object from the hands of the patients because of this finger flexor affection, and also by the difficulties of climbing stairs of affected patients. This disease is progressive as well as Parkinson, leading patients to high degrees of dependency. Usually they are finally related or restricted to wheelchair mobility. And they also experience serious problems of dysphagia, leading to difficulties in food intake. At molecular or pathological level, this disease that is diagnosed still today by muscle biopsy includes the presence of three features in the muscle biopsy. First, the inflammation that is characterized by the infiltration of T lymphocytes, specifically the CD8 positive T lymphocytes, as well as the presence of major histocompatibility complex 1 antigens. in the muscle cells. This is the first feature to diagnose the disease at pathological level, but also we have to observe degeneration that is present through the formation of remed vacuoles that store unfolded proteins. Which unfolded proteins? The same as they are always present in all neurologic and neuromuscular diseases. That means beta-amyloid, caveolin, TDP43, P62, tau phosphorylated, and all these proteins that are abnormally big, and probably because of that trend to be unfolded in all these neurological diseases. We also have to observe mitochondrial dysfunction, and this is the first metabolic sign of the disease I was telling you that we could be observing in both Parkinson and IBM. In the case of IBM, these mitochondrial disturbances are pathognomonic signs to diagnose the disease, and they are expressed through the presence of ragged red fibers that are related to the abnormal proliferation of mitochondria in muscle cells, probably to compensate mitochondrial dysfunction. Mitochondria trend to increase their number, and also by the presence of COX-negative SADH positive cells, that these are clinical features typical of primary mitochondrial diseases, and they are also common to IBM. Parkinson's disease also show mitochondrial dysfunction, and we will later talk about this. Both PD and IBM also share other similarities. For instance, they are age-associated disorders, and they are manifested or prevalently manifested at advanced phases of life. Usually they start at the age of 60 or more. And despite their prevalence is highly different, is much more prevalent in Parkinson than in AVM, the number of patients affected by both diseases is highly relevant worldwide. They show or they share the socioeconomical burden, the number of patients affected, that they are age-related, and also the molecular basis, because in both cases it has been hypothesized or related the presence of all these molecular features for both diseases: inflammation, oxidative stress, proteostasis deregulation, ER stress, the presence of protein depots, as said before, the presence of autophagic imbalance, calcium buffering deregulation or distress, and especially mitochondrial dysfunction and metabolic reprogramming. In both diseases, there are genetic and sporadic or idiopathic forms of the disease. The idiopathic forms are still the more frequent or the more prevalent forms of these diseases. But as long as new generation sequencing advances, this percentage of idiopathic forms will be reduced. In the case of Parkinson, a lot or frequent forms of the genetic forms of the disease are related to mitochondrial function, since they are genes encoded in the nuclear genome, but coding for mitochondrial proteins that are responsible of different mitochondrial functions. But most of them, like Parkin, PIN1, DJ1, etc., related to mitochondrial dynamics, that is mitochondrial function, fusion and fission. This process allows mitochondria to exchange genetic and structural material to renewal of the internal mechanisms of mitochondrial function. In the case of IBM, the genetic form of the disease is not directly related to mitochondria because it's related mainly to glycosylation. But in the case of the idiopathic form, it has been, as said before, highly related to pathological mitochondrial disturbances. And we have also described molecular alterations, for for instance, in the presence of empty DNA deletions, that are deletions in the mitochondrial genome, and also the production of ROS or abnormal mitochondrial dynamic fusion and fission, and also how all these mitochondrial disturbances are related to the triggering or promotion of cell inflammation from inside the cytoplasm. These diseases, as said before, share a lot of similarities: the sociosanitary burden, the huge prevalence, the molecular basis, and also the research gap and clinical needs, because in none of them it's known the primary mechanism that triggers the mechanism of cell disease that leads to the dysfunction. There are not well-known non-invasive biomarkers for none of these diseases. Parkinson's disease is diagnosed mainly through clinical signs, and IBM through clinical signs and muscle biopsy. And finally, none of them have therapeutic strategies, at least at long term, because Parkinson, lucky us, has L-DOPA treatment, but at late stages, sometimes there is resistance to the treatment that needs to increase doses and be directly targeted to the central nervous system. And also there are secondary effects of this treatment. So we need to develop new therapeutic strategies, and hopefully curative and preventive, that can be administered at first stages of manifestation of this disease. And to develop all these things, we need models to study these diseases. There are a lot, but we need still to improve in the development of new cell models that recapitulate the main hallmarks of the disease. Usually in biomedicine, as you know, we work with animals or cell lines. In the case of animals, we usually develop the cell models by toxic exposition or gene editing. In the case of PD or IBM, this idea wouldn't be much related to the natural trigger of the disease, because there is no direct association with toxic exposition for the generation or the development of none of these diseases. And in the case of IBM, the genetic editing wouldn't be related to the idiopathic form, neither in the case of Parkinson. What about cell lines? They are useful and have low maintenance and facility costs. However, they are cancerous. So there are two typical features of these cells, like high replication rate and prevalent glycolytic metabolism, that quietly differ or is different from the target tissue of both Parkinson's disease and IBM, because both neurons and muscle are tissues of post-mitotic origin that do not replicate themselves, at least in high rates. And additionally, they show oxidative metabolism. So maybe they wouldn't be the best model to recapitulate or to study these diseases, despite they also show good approaches for some studies. That's why we planned, or we hypothesized, that fibroblasts could be an interesting model to study both diseases, at least fibroblasts derived from patients, because they conserve the genetic, epigenetic, and environmental cues of affected patients. They also show some disadvantages, because they still show some renewal rate. I mean, they divide, not as much as cancerous cell lines, but they still divide. And they still show some rate of glycolytic metabolism, not as much as cancerous cell lines that mainly rely on glycolysis, but they have kind of mixed metabolism, oxidative and glycolytic. They also can be, contaminated by mycoplasma or other conditions in the culture room, and they have to be strictly controlled to be between passage 3 and 10 because you have to avoid keratinocytes that are present in lower passages, and you have to be close to the patient to avoid potential disturbances in passage above 10. But once you take care of these things and you are conscious of these things, they offer interesting advantages like facility costs that are low. They are they do not suppose an invasive approach for the patient, and they can be, for instance, transdifferentiated into the target tissue, either muscle or neurons, or then derived into IPS to further be differentiated into this target tissue. So they are interesting. And by their own, as fibroblasts, they offer by themselves the possibility to recapitulate the diseases. And that's what we will try to show you today, that these are interesting cell models. And in the meanwhile that we validate this model of disease, we will unravel molecular players that can be used to better understand the etiology and even postulate candidate or surrogate biomarkers of the disease, and even speculate of potential therapeutic targets. So we will focus this speech in the validation of the cell models, either by functional or omic techniques, as well as in the description of molecular key players we think that may be interesting to better understand the basis of the disease. We will work with fibroblasts, as I said before, extracted by punch biopsy from the patients that suppose a minimal invasive approach. We will work with these sample size cohorts of 6 to 8 PD patients that harbor the mutation in Parkinson gene. That is a mitochondrial related gene that controls the fusion and fission of mitochondria in the mitochondrial dynamics process. And we will compare these results with respect to 12 controls. And in the case of ABM, we have a bigger sample size of 14 to 15 individuals, and we will also compare to the same controls. In these fibroblasts, we will do the functional analysis or phenotyping of stunning mitochondrial function, inflammation, and degeneration, because of the main features in IBM, but also in Parkinson. And we will also perform an unbiased and untargeted approach by transcriptomic and metabolomic techniques. Let's move to the results section, and we will talk first about the functional validation of these cell models, and we will start with bioenergetics or mitochondria. In the case of PD, you will see patients in black bars, and you will observe that they showed less or decreased basal respiration, as well as complex 1 oxidative and enzymatic activity, and also higher rates of oxidative stress. In the case of IBM, we observe also decreased respiration or oxygen consumption, that in this case, patients are graphicated in white bars. Take care with this, is to maintain you awake. In this case, as said before, in ABM patients, patients are graphicated in white bars and show less basal respiration, less ATP production, a decreased mitochondrial membrane potential that is highly related to apoptosis, a higher lactate excretion or secretion that is highly related to mitochondrial diseases, because when pyruvate cannot enter into the mitochondria, is conducted through fermentation or anaerobic glycolysis to the production of lactate. And also we observe in patients the regulated reagent or ROS production. What happened in the functional validation of the inflammation? In this case, we only have results of IBM patients because the Parkinson's disease study was performed before, before, and we didn't realize we could do this analysis, but probably we would observe a similar pattern. To measure inflammation, we quantified the presence of secreted cytokines into the supernatant of the fibroblast cultures, and we observed that most of cytokines were upregulated in these supernatants, as you can observe by hot or cálid colors in red. Most of the cytokines were upregulated in fibroblasts of IBM, especially eotaxin, that is related to eosinophil stimuli, granulocyte colony stimulating factor, as well as monocyte chemiotractant protein 1. But these results, I think we need to point out that fibroblasts are not cells of the immune system, and because of that, the presence or the secretion of these cytokines is highly relevant, and they must be really inflamed to secretate these molecules into the supernatant. We also characterized degeneration in these cells, and to do so, we measured the presence of autophagy, that, as you know, is the process cells activate to clean, to eradicate any cell debris that can be, for instance, unfolded proteins, but also dysfunctional organelles in the cell that are not working, and the cell activates this autophagy to get rid of these structures that interfere with cell homeostasis. And to measure autophagy, we perform a time course experiment by measuring one of the proteins that starts this autophagy, that is P62, that targets all the debris, all the things that need to be cleaned, and LC3B2, that is a protein that finishes this autophagy, but by closing the autophagosome after the lipidation. So we measure one protein at the beginning and at the end of the process, and we observe that PD patients in black bars show a decreased level of expression of both the starting and the ending proteins of the process, the markers P62 and LC3B2. In the case of IBM, we observe the same pattern. In this case, again, take care, the patient are graphicated in white bars, and we observe that the same pattern of reduced expression of these markers was present in IBM patient, but maybe less accentuated, less pronunciated. But in any case, it was significant in the case of LC3B2. These levels were reduced, suggesting that autophagy was downwarded, was reduced in these fibroblasts, together with an increased inflammation and deregulated mitochondrial function. The decrease of autophagic proteins was not only restricted to P62 and LC3B2, but also was downregulated for most of the proteins taking part of the macroautophagy and mitophagy process. So we concluded that this global process was downregulated in IBM cells. We also could observe less presence of autophagosome formation in IBM fibroblasts with respect to controls, suggesting that this process was downregulated. And we also observed higher presence of abnormal lysosomal and fusion without lysosome structures in these cells by electron microscopy. We also observed that IBM cells showed mitochondria more dysfunctional, at least morphologically, and we previously studied or measured the dysfunction of mitochondria. Mitochondria are imbalanced or altered not only at morphological level, but also at functional level. So the conclusion of all these functional assays is that fibroblasts show a differential phenotype between cases and controls in both diseases, confirming the validity of the fibroblast model. But we also wanted to assess the validity of this model with a non-targeted hypothesis, not looking for the features that were altered in the target tissue of the disease, but also an omic approach in which we could observe any other alteration in pathways non-directly or non-primary related to the disease. And we started with the transcriptomic analysis of this fibroblast. In the case of Parkinson's disease patients, when we perform the principal component analysis, we observed that they clustered together, patients by one side and controls by the other, splitting, and this was a good news. It means that in an unbiased analysis, patients were more closely related between them than with respect to controls. When we later on performed the algorithm and filter analysis, we set up the signification of differential expressed genes at 0.05 fold discovery rate, and the fold change between up or down regulated gene between cases and controls in two units. When we do so, this fold discovery rate of 0.05 and the fold change at 2 units, we observed 343 deregulated expressed genes, 206 upregulated, and 137 downregulated. And again, this is highly relevant because we are talking about one fibroblast in the skin of one patient that do not show clinical differences with respect to the control. I mean, it's not a neuron, it's just a fibroblast of the skin, but it shows 343 deregulated expressed genes with respect to a control. Which pathways are deregulated? These 343 genes belonged mainly to pathways of cell adhesion, cell growth, but also metabolism, especially the one restricted or related to amino acids and to folate. What about IBM? In the case of IBM, we also observed clustered gathering or differential association of patients with respect to controls. And again, these were good news. There was a clear differentiated pattern of expression of patients with respect to controls. And in the case of setting the same requirements, meaning FDR at 0.05 and 2-fold change expression levels of difference between cases and controls, we observed that there were 778 deregulated expressed genes in this IBM fibroblast related to pathways of RNA processing, cell communication, but also amino acid metabolism. That's why when we performed the metabolomic analysis, we targeted the organic acids and the amino acid metabolism, trying to validate what we observed previously in the transcriptomic analysis. Both amino acid and organic acid metabolism is related, as you can see in this diagram or in this figure. And both are related also to bioenergetic functions. In the case of Parkinson's disease, due to Parkin mutations, we observed that most of the amino acids were upregulated, as you can see in colored colors or in red. They show a global pattern of upregulation with some exceptions, but most of the amino acids were upregulated. And this is usually related to the activation of the mitochondrial induced stress response. I don't know if you heard about it, but it's a nuclear effector response that is activated when there is a mitochondrial lesion. It has been widely described in the case of primary mitochondrial diseases. Let's imagine that there is a mutation in the mitochondrial genome, and then the nucleus to counterpart to try to compensate this mitochondrial dysfunction activates a series of pathways trying to counterpart this lesion. For instance, there is the activation of the antioxidant metabolism through glutathione expression and activation, but also in mTOR1 activation, transsulfuration, the expression or the increase in NADH pools to increase all the anabolic efforts of the cell, and also the increase in amino acid expression to promote protein creation and synthesis. And probably that's what we are observing in this neurological disease for the first time, because it was described in case of mitochondrial diseases, but not in the case of a neurological disease like Parkinson. This response was clearly associated to folate metabolism and 1-carbon cycle metabolism, and we could observe the presence of increased folate synthesis, probably trying to feed all the pathways related to cell cycle, to antioxidant response, and also the methyl donation needed to do all the epigenetic changes that take care of or take account in this disease. Interestingly, when we measured the presence of these amino acids in liquid biopsy, meaning non-invasive fluids, biological fluids of the patients, in this case in plasma of the patients, we observed that some of these amino acids or the combination of some of these amino acids led to higher expression in patients than in controls. We have to explore this in a big sample size, but it suggests that these amino acids, the regulated, could be used as diagnostic or even prognostic biomarkers, at least in Parkinson PD disease. In the case of IBM, we performed the parallel analysis of amino acids and organic acids. You can see in the left-hand side panel the presence or the expression of amino acids, and as you can see in this color graph, there are not a clear pattern of up or down regulation of these amino acids. We observed that some of them were conserved in white color, some of them were down-regulated in blue color, and some of them were upregulated in red. But there was not a clear pattern as observed previously in the case of PD. However, in the case of organic acids, we observed this upregulated pattern of expression in IBM, as you can see in these colored or red colors. And when we analyzed specifically some of these organic acids, like lactic acid or 2-hydroxyl glutaric acid or pyroglutamic acid, we observed that these differences were significant. Again, when we planned to measure these organic acids in non-invasive fluids, in this case in urine of patients, we observe again the expression of these organic acids in urine, suggesting again the presence of surrogate biomarkers of the disease outside of the fibroblasts in other biologic fluids that could be used in or for a non-invasive approach. So these are the conclusions of this speech. I hope to convince you at that moment of the speech that fibroblasts recapitulate the main hallmarks of PD and IBM, both at functional and omic level. And also, and these are conclusions derived from the first one, that these diseases are more systemic than initially thought. Oh, we thought that they were restricted to neurons and muscle, and no, they are spread or present in other cells and tissues of our body. They are also diseases more associated to the metabolic reprogramming and bioenergetic imbalance than initially thought. And we want to point out that the study of these fibroblasts can be used to better understand the disease in etiologic means, but also in terms to promote biomarkers or even develop or study new therapeutic targets for the disease, maybe by trying to regulate these imbalanced metabolites that could be taking care or being part of the etiologic mechanism of the disease. And nothing else. I wouldn't like to finish without thanking my colleagues at the Muscle Research and Mitochondrial Function Laboratory. If fibroblasts resemble PD and IBM, they resemble, at least to me, all the heroes from any Marvel film. And also, I wouldn't like to finish without thanking all the collaborators that take part of this study, specifically the neurology department in the Hospital Clínic Barcelona that provided the PD patients, because since we are multidisciplinary group, we also have patients that provide us with the neuromuscular patients. But to study PD, we need the neurology department of our hospital. And I also would like to thank the Inborn Errors of Metabolism Laboratory at Hospital Sant Joan de Déu, led by Rafa Artuch, that allowed or facilitated the metabolomic study and all the assessment of this deregulated metabolism that we are facing in these studies. Nothing else. Thank you so much for your attention.
Thank you, Glòria. I only want to remind you that you can ask your questions in the chat, both in the YouTube and also in the Zoom chat. Glòria, thank you for your talk. It has
been a pleasure.
Fantastic. At this moment, we don't have questions in the chat, but I have many questions for you. And I suppose that Josep has also many questions to start.
Questions for Glòria too. Yeah. So I can start. First of all, thank you, Glòria, for this wonderful talk, this enlightening.
My pleasure.
Talk. I was actually not familiar with those inclusion body myocytes. So, and I found very interesting the fact that when you measure about the cellular features of this disease, one of them is the inclusion of some proteins that are the same as you can find in patients with Parkinson's disease. So I don't know what that means, that the etiology or the origin of this disease is kind of similar or not. But my question is whether you know if patients, for example, with Parkinson, do actually show some kind of signs of myositis anytime along the course of the disease, and the other way around, whether people with a clear muscle manifestation of this disease, myositis, at some point they also show some kind of neuronal degeneration or neural defects.
Mm-hmm. Really good point. This one, Pep. To start by any point. This clinical or molecular, more than clinical, these molecular features that share both diseases are typically associated to aging, and probably these diseases are accelerated aging of specific tissues affected. But the protein unfolding and the inflammation and the oxidative stress and metabolic reprogramming probably is typical of any disease associated to aging. The point is that, in case of IBM, there is the presence of proteins depot as well as in Parkinson. And despite the composition of the protein depots is slightly different, in the case of Parkinson, there is more alpha-synuclein, for instance. And in the case of IBM, there is more beta-amyloid. Probably the mechanism of protein unfolding and storage is the same. In fact, IBM is called the Alzheimer of the muscle because it's the same remnant vacuoles that are present in myofibers of IBM patients are present in neurons of Alzheimer's disease patients. So probably we are talking about molecular feature present in many diseases associated to aging. And specifically, we performed a clinical study with a small cohort of 20 patients with IBM, if I'm not wrong, in which we tested the presence of Alzheimer, and it was not increased with respect age and gender paired controls. However, in case of Parkinson, there is kind of muscle wasting. The point is, it is due to molecular alterations, or it is due to movement impairment present in these patients that leads them to a sedentary lifestyle. Probably we need to research more in this, but it is really an interesting point because, as said along the speech, there are common similarities between all these entities, and probably there are common mechanisms, not only clinical signs, but also mechanisms, molecular mechanism underlying all these comorbidity.
If I can just follow, sorry, Mar, just following with this the previous question. I mean, when you describe the cellular alterations, you just mentioned a lot of things that are going on in these cells, like, you know, the accumulation of certain kind of proteins, mitochondrial dysfunction, inflammation, and at least for all of the people like me that are not familiar with neurodegenerative diseases, I mean, we used to think that Parkinson, for example, is just a result of a loss of dopaminergic neurons as a result of accumulation of these tau or synuclein proteins. So it appears that there are a lot of things that might be going on in those cells that are originating the disease. What can you tell us about the real or the primary cause of this disease?
Yeah, I guess nobody knows which is the primary cause of the disease, and that's why we are here. But my feeling, I mean, it's not a consensuated theory or something, but my feeling is that probably this disease, as well as IBM, I mean, Parkinson, but also IBM, are the finally manifestation of different molecular triggers. So you can start by one alteration, either functional or genetic, and one follows the other. Maybe if you don't, I don't know, you don't properly folder your proteins, you will have some inflammation, and then it leads to an abnormal metabolic reprogramming, and then to oxidative stress, and then finally you lose your neuron or your myoblast, your myocyte, sorry, in both diseases. But you can start by another point. You can start maybe by increased oxidative stress due to abnormal mitochondrial function that leads to inflammation, and finally to normal proteostasis and protein deposits. So probably there are different genetic and molecular triggers that can lead to common cell dysfunction and clinical manifestations. So probably when we clinically categorize or try to establish the name of the disease, we are giving the same name to different molecular and genetic entities. And I think that as long as we advance in the research, and for instance, in genetic testing of these patients with this new generation and omics approaches, probably we will realize of the complexity of these diseases that we target with the same name.
Okay. We have a couple of questions from the audience. Susana Cadenas says, very interesting talk, Glòria. Does the decrease in CI activity in Parkinson's disease have any effect on mitochondrial ROS production? Would a decrease in ROS generation alleviate or delay the symptoms?
Sorry, about the first question?
Does the decrease in CI activity? I don't know if you know exactly.
Complex 1 mitochondrial activity, I guess.
OPA1?
OPA1?
I don't know, Susana, maybe you can tell us what do you mean with CI activity?
Or maybe I saw the question written.
She's referring to mitochondrial complex 1.
Complex 1. Okay. Yes,
if this
probably
decrease in complex 1 activity is related, sorry, to?
mitochondrial
ROS production.
Yeah, absolutely, absolutely. ROS, I mean, reactive oxygen species, is the responsible of cell damage and is partially compensated by the antioxidant defenses, but they are mainly produced. 90% of ROS stress is produced in mitochondria, and is highly related to mitochondrial respiratory chain function, and especially to complex 1 and 3. So complex 1 dysfunction is highly related to ROS production, and despite the nucleus activates this induced stress response I was explaining you that is classical of primary mitochondrial diseases, if the ROS levels are too high, despite the nucleus activates the induced stress response, there is no possible correction or compensation, and cell damage accounts. So yes, it's highly related to complex 1 dysfunction.
Yeah, Susanna, sorry, Susanna was also asking whether if you can somehow decrease RS production, you will actually alleviate or delay the development of the pathology.
I think so. I think so. And when we have tested it in animal or cell models, we have kind of decrease the speed, or I don't know how to say, decrease the progression of the disease. Despite we were talking about, maybe it's not the trigger, the first molecular player that started the disease, but if you are able to compensate any of the downstream down regulations, maybe you can stop the disease. That's why we are interested in mitochondria, because it can be the origin in case of some genetic Parkinson, as the one we were explaining today, in case of Parkinson. But even in the case of other forms of the disease where genetic mutations are not directly related to mitochondria, mitochondria can still play a role on this. And if you are able to ameliorate mitochondrial homeostasis by decreasing ROS production, for instance, maybe you are able also to decrease the level of manifestations of the disease, or retard, delay the expression of symptoms or the severity of these symptoms.
Okay, thank you. We have also a couple of questions in the YouTube chat. Matthias Staras says, great talk. Is there any way to validate these transcription chains and metabolic reprogramming that you found in fibroblasts in the IVM patient's muscle tissue?
If I understood correctly the question, this question is related to the demonstration of the
metabolic changes that you find in your fibroblast. If you can think that you are being able to validate them in the muscle from the IVM.
Okay, I get it. Thank you, Mar, for the translation and explanation. That's we are focused on that because we started first by searching or validating this model of a study by looking for the alteration of muscle in fibroblasts, but then we have to do the back way around and looking for the alteration we have found in the transcriptome or any other parameter we assessed in these fibroblasts, but in muscle. Probably there will be differences. I mean, probably there will be alteration that will be specifically of the tissue we are studying, but some of them will be common. And that's why we are interested in this cell model directly derived from patient, because it still conserves a lot of the regulation present in the target tissue. But probably some of them will be just restricted to muscle or just restricted to fibroblasts.
So Ruth Enriquez also says that thank you so much and congratulations for this webinar, Glòria. I'd like to know if these biomarkers are present in those patients with mild Parkinson's disease, or if they could be useful to give as a hint that the disease could be developing in those with some kind of genetical predispositions to suffer Parkinson's disease.
I guess that she's referring to the presence of surrogate biomarkers in non-invasive fluids like urine or plasma. And this is a really recent finding of the last weeks. I mean, in Christmas times, we were analyzing this data, highly excited. And we have to deeper explore. But by the moment we can assure that they are present in Parkinson PD. We have to look at other kind of Parkinson, like the idiopathic form, that is the more prevalent one. Okay. And in the case of IBM, we could look at other neuromuscular disease and try to understand if it's specific of IBM or can be exported or translated to other neuromuscular diseases.
And I have a curiosity. Have you tried to restore the Parkin mutation to the wild type is it possible in the patient fibroblasts and to see if with changes are restored? Just to know if this is an initiating point or not.
Yeah, we haven't done it, but this is typical when you do animal models or cell models, no? The rescue experiments. In patients is not that usual, but it can be done to make sure that there are no complementary mutations or any other comorbidity that could be also interfering or
contributing,
contributing or giving complexity to this phenotype. Yeah, a really interesting point.
Okay, so Josep, if you have more questions, I think that we have Glòria has answered all the questions from the chat.
There are no more questions on the chats, but I do have one or two more questions, so depending on the time. So when you describe the symptoms of Parkinson, there was something that caught my attention, is that Parkinson patients actually experience some excessive sweating. And as you know, thermoregulation is one of my major topic of research. And I was wondering, how's that? What could be the cause of this excessive sweating? I guess that patients or the body of these patients feel like need to cool down or to thermoregulate somehow. No? That is response of an excessive sense of temperature. I don't know. What can you tell us about?
Yeah, Pep, probably you can talk about this longer than me because I only know the
not about Parkinson
clinical signs. They are prodromals. I mean, this is very interesting because they are at the beginning of the disease, like the sleep disturbances. They are called rapid eye movement sleep disturbances, and patients move a lot in the bed, and these are the first stages. So probably there's the trigger or the molecular trigger is taking account of these clinical signs and symptoms that appear at these prodromal states. And the issue about sweating, I don't know if it's related to the autonomous nervous system affection that maybe accounts before that the central nervous affection, or as you said, is a kind of need to thermoregulate. For instance, if mitochondrial is implicated in this disease, as you know, mitochondrial consume all the substrates from the cell to produce either energy or heat. So maybe if mitochondria is affected, there is a kind of, I don't know, thermoregulation problem, and this is why patients tend to sweat in abnormal rates. Maybe Pep, you can help us with all this. And
I'm not sure, but I mean, what you're suggesting is that perhaps those mitochondria are more uncoupled. I mean, even if they have a reduced production of ATP, that's in part because of they're uncoupled and they produce more heat. So they are working basically inefficiently. Yeah, that's an interesting.
It's an hypothesis. I mean, I don't know if it's just a question that primary is affected, the autonomous system regulated these autonomous functions, or there is a molecular explanation of mitochondrial and thermogenesis deregulation.
Okay. So I was wondering whether beyond the dopaminergic neurodegeneration, there are other neurons in those, nuclei that are also being affected by the disease, and that can explain, the defects in thermoregulation and many other, the regulation of many other physiological functions of the body. So
yeah, it's believed, or it's primary thought, that dopaminergic neurons are the main affected in the disease, and it's supposed to be quite specific, but probably other alteration. I think it has been speculated, some GABAergic and some other neuron affections. So yeah, probably again there is a complexity of molecular things undergoing there. We only know the point of the the upper part of the iceberg.
We have another question for Victor Alvaro. He says, thank you so much for the webinar. My question is if you know if the molecular changes that you observe in fibroblasts occur first or at the same time as the molecular changes in the target cells of the diseases, neurons or myocytes.
Yeah, my belief, and again, this is a belief, it's not a consolidated theory, is that probably these disarranges take place at the same time, but in the case of postmitotic tissues like Parkinson disease and IBM primary affection, meaning neurons and muscle cells, since they do not have any replication rate or really slow, and really small, and they cannot have any kind of renewal, these alterations get stored in these cells. I mean, probably a blood cell that is not a cell memory living 20 years, but is exchanged each 20 days, it's less difficult that they show these molecular alterations with respect to a neuron or a muscle. And in the middle, probably there are the fibroblasts, these cells that are quietly renovated, but they still live longer enough to show these disturbances. But my guess is that probably these disturbances appear at the same time everywhere, and some tissues trend to show more deregulation because of this slow and small cell renewal.
Okay, I think that we have no more questions. So if you don't have more questions, Josep, I think that it's time to finish.
Yeah. I don't. Well, I mean, I have questions, but I think maybe leave it here. So I just want to, thanks first of all the audience for attending this webinar, and I hope that we'll be connected to the next webinars, and maybe see us at the in vivo in live at the next meeting of the SEBBM in Malaga. I would also like to thank Mar for co-hosting and helping us with technical issues of this webinar, and of course I would like to thank Glòria for this wonderful talk.
Thank you so much.
Thank you, and see you next time.
Okay, thank you very much to both of you, Josep and Glòria, and for the fantastic talk, and also for the discussion that has been really interesting.
Yes,
you can stay if you want a little bit in the Zoom discussing, but we are going to close the emission now. Thank you very much again, and also thank you for the audience.
Hope to see you in person next time, all of you.
Ahora ya estamos.